Yes.
When should we know the result?
Right after this.
Oh, okay.
Depends how fast I can grade. By 1? All right. As I said before, I'm usually going to put the first, slide on here as these are some prefixes, root words, things like that you'll see throughout this PowerPoint that I may go back and refer to. Again, a lot of times if you know what the word means, it tells you exactly what it does. So it becomes helpful. So I have no idea how far we'll get. We will not be finishing this PowerPoint today. But we'll start it. All about the cells. It's a very important unit because we'll talk about basic cells in every single unit after this. When we start about any unit, we have to know what kind of cells are in it. Now, all cells have some common structures and functions. And then, yes, they do diversify depending on what body systems and what they need to do. They all have 3 basic parts. They have an outside that closes everything in. That's the plasma membrane. That's the boundary. They have the cytoplasm. This is the fluid on the inside that has all the organelles, whatever they might be, for that particular cell. And they all have a nucleus. They all have their control center, their main organelle that contains all the genetic information. So all cells, no matter where you are in the body, do have at least these 3 basic parts. Outside, fluidy stuff inside, and they all have a nucleus. This is like your big, basic picture of a generalized cell. And I'm like, we already looked at a lot of these structures and I'm like, lab already, so some of this should be review. Yay. So I'm not going to spend a lot of time on that big, huge picture labeled. It's in your book, too. Now, on the outside, the plasma membrane that holds everything in, it is a lipid bilayer. What does the bi part mean in bilayer? 2. It's a double layer. And we'll look more at what makes it all up in a minute. And I'm like, it has lipids, yes, and it has proteins. And they are always changing on what is where. It's called, some people call this the fluid mosaic model. Fluid, because they're moving. Mosaic means changing. And so they're changing as they move around. So I tried to find at least a little tiny, image. And, if this purple proteins are always purple blobs, wants to move over there, it can. It can just shift its way over there. Things are not set in place in the plasma membrane. Stuff is shifting all around. It's kind of like our planet. We're all on our own little continents, but we're all on a fluid magma. We're shifting. Same thing. They're all shifting around too. Now, it does separate the outside of the cell from the inside of the cell, and there's a fluid on both sides. The inside fluid we call intracellular fluid. It's our cytoplasm. Intra, what do you think intra means? Inside. It's our intracellular fluid. And then extracellular fluid, extra means outside. The outside fluid, about 20% of our extracellular fluid, the fluid that's outside the cell, but 20% is plasma, and the other 80% is interstitial fluid, the fluid found in between cells. Inter means between, and I'm like, so it's the fluid found in between cells. So it's dividing the fluid from the outside from the fluid from the inside. They are slightly different from each other. This is a nice picture of the plasma membrane. Here's that phospholipid bilayer. There's all your extracellular fluid on the outside, and your plasma or your cytoplasm or your intracellular fluid on the inside. It divides the two. Now, we find lipids in there, and about 75% of the lipids that are in that phospholipid bilayer are phospholipids, hence the name phospholipid bilayer. So about 75%. Now, a phospholipid, the general drawing of a phospholipid is a head with 2 squiggly tails. They draw it a couple other ways depending on, how you want to draw it out, the little bubble models or lines and looking at bonds, don't care. Head with 2 squiggly tails. Now, the top part is a phosphate head. That's where we get the phospholipid. And the bottom part are our fatty acid tails. Now, our phosphate group is polar and hydrophilic. Now, hydrophilic. I think I've got these on the first slide. What's hydro referring to? Water. What's philic mean? Loving. It's the opposite. And I'm like, so hydrophilic, what does it mean? It's water loving. It loves water. So this phosphate head hangs out where there's a watery solution, like the extracellular fluid or the intracellular fluid. It likes water. Now, things that like water are also called polar bear, or polar, and I always think of it because if you've ever gone to a zoo that has a polar bear, do polar bears like to swim? All the time, they love swimming. And so polar bears like water. Things that are polar like water. Now, fatty tails are nonpolar and hydrophobic, which means what? They hate water. Like they want nothing to do with water. However, extracellular fluid and intracellular fluid are both watery types of solution. So do they like to hang out where there's water? No, because they're lipids, and I'm like, they're the kind of the lipid portion of it. This is why when we put a whole bunch of these phospholipids together, they form a bilayer where the fatty acid tails hang out together, fatty acid tails, and those phosphate heads hang out on the edges because there's a watery solution on this side and there's a watery solution on this side. And these tails, they figured out a way to be the farthest away from the water as possible is if they huddle together on the inside. So they form this bilayer. Now, some other lipids that we find in our membrane, we have glycolipids. What do you think the glyco refers to? I heard it. Say it. Sugar, yes, and I'm like, it's a lipid with a sugar attached to it. If I go back, I was going to say, there's a lipid with a sugar attached to it, glycolipid, it just means sugar lipid. And about 20% of our lipids we find in our membrane are cholesterol. It's going to be like coughing on my poor little microphone here. Now, the cholesterol we find inside the membrane. Now, cholesterol is a lipid. There's no watery portion to it. So it hangs out by the lipids. The lipids like the lipids. So it hangs out with the tail region. So it's going to be found inside the membrane. What it's going to do is increase membrane stability. This is fluid. Again, if this particular protein wants to move over there, it can. It's fluid. However, we don't want our membrane to be too fluid. And so by having these cholesterol molecules there, it kind of firms up the membrane, makes it a little less fluid, a little more stable. I was going to say, here was my next picture with the circle. But you can see all of the little the phosphate heads, the tails like to hang out together, and you have phosphate heads on one side, phosphate heads on the other side. Also in our membrane, it's like the more I talk, the more I cough. Also in the membrane, we have proteins, purple blobs. And there are two types, integral and peripheral. Integral proteins mean they go through the entire membrane itself. They even gave me a purple. Meaning, I'm like, I have a protein that goes through the entire membrane. So it peeks out on either side, just like those three do. I'm like, they peek out on either side. They're also called transmembrane. Trans means across. If you ever have to take a transatlantic boat ride, it means you go across the Atlantic. It just means across. It goes across the membrane. It's a transmembrane protein. Now, because they hang out in an area where you have polar heads, you know, and I'm like, and because they have to hang out with these fatty acid tails, the proteins themselves have hydrophobic regions and hydrophilic regions so that they can insert themselves. And again, the hydrophobic region likes to hang out with the hydrophobics. And the hydrophilic regions like to hang out with the hydrophilic heads. And so the protein itself, changes a little bit in its own structure just so it can fit in there and stay in there. Now, if they go all the way across, one of their biggest function is they are transport proteins. Whether they are a channel that just stays open all the time, or they're a carrier, meaning they're going to physically pick something up and push it to the other side of the membrane. And I'm like, they're there for transport, for stuff to go across the membrane. They can also be used as enzymes, because enzymes are proteins, or they could be there as a receptor, trying to receive information. Imagine this is like, a little antenna up here, and it's like, I'm receiving information that I'm going to tell inside the cell. Peripheral proteins do not go all the way across. Peripheral means kind of on the outsides. I know I've got a picture. Here is a peripheral protein. And I'm like, it kind of hangs out on the edges. And I'm like, it doesn't go all the way into the cell. But it still is part of the membrane, but it's just not inside the membrane. Now what they do, and you're like, but it's not in the membrane, it's not part of it. It's attached to it though. They can function as enzymes. They can help change the shape of the cell, especially when it's starting to divide and it needs to pinch off and pull apart. It can help during muscle contraction, changing the shape of the cell. And it can help cells stick to each other. And cells want to connect. So you've got different, they're all proteins. They do their own little thing, and they're kind of found whether in the cell or in the membrane or on the edges of the membrane. Now, here are just kind of some, I've already mentioned some of these, but if you are a protein found in the membrane, you have 6 functions. You could be transport. It's usually your transmembrane ones that go all the way across. And I'm like, so you're moving things, whether, red circles or whatever it is. They could be receptors. And so this particular protein is receiving information from the signal, whatever that little wedge thing is. When it receives the signal, it sends information inside the cell and will cause some type of chemical reaction to happen. It's kind of like, okay, I'm just waiting for the receptor coming along, then I'm going to stimulate some other type of chemical reaction in the cell. They can be attached to the cytoskeleton. The cytoskeleton can give some shape to the cell, just like our skeleton does. And I'm like, but the cytoskeleton likes to attach to something, and it can attach to some of these membranes to help give the cell its shape. It could be enzymes helping some type of chemical reaction. There's 2 of them working together. One turns green circles into red triangles, and one takes red triangles and turns it into blue squares, whatever they are supposed to be. But they're helping speed up chemical reactions. They can help join 2 different cells together. Proteins can stick to proteins. Or they can help in cell recognition. Some proteins, glycoproteins, which just means sugar protein. It's a protein, sugar molecule attached to it. I'm going to talk in one of my next slides about these sugars. But I'm like, these sugar particles attached to the protein, the sugar, the carbohydrate portion of it, is distinct. And I'm like, what the structure looks like, how many it has, it's distinct for the cell. And so once this cell is like, well, I'd like to attach to whatever kind of cell, it can recognize this cell based on the structure of that carbohydrate attached to the protein. And then it's like, you're the cell I need to attach to, and it'll stick to it. So they can recognize each other based on these little carbohydrates stuck to the proteins. Here's my sugar. I knew it was coming up. And so we find these sugar carbohydrates, and I'm like, these sugar coverings only on the cell surface. We do not find these inside the cell along the cell membrane. They're only at the cell surface. They're attached to lipids. They're attached to proteins. And yes, we usually call them a glycolipid if it's attached to a lipid, or a glycoprotein if it's attached to a protein. Every cell has a different pattern of sugars, They allow for cell-to-cell recognition for one cell if it wants to attach to another one. And it allows our body, especially our immune system, to recognize cells that are our own versus cells that are foreign. So we can start recognizing bacteria, and we can recognize other cells that might be foreign. Be like, we don't know what that is, so we better just get rid of it. And so it can recognize anything that's our own, just because of these sugar coverings on the outside of the cells. Now, the cells, as I said before, on the outside, they're surrounded by mostly interstitial fluid. And interstitial fluid on the outside, it is a sugary environment. Things you can find in it, other than it's water-based. And I'm like, it's not sugary. I'm like, it's a watery environment. We can find amino acids, sugars, fatty acids, vitamins, hormones, salts, waste products. You know, it's just a water with a whole bunch of things dissolved in it. But that's fine. You have this watery substance on the outside of the cell that has all of these different things in it. Our cell can figure out what it needs and what it will allow in. You know, if our cell's like, oh, I need some vitamins, it can try to move some of the vitamins inside the cell. It's like, I need some amino acids because I'm building some proteins. It can get some of the amino acids in. And so it's almost like having this fluid outside of the cells that just has like this bank of nutrients for whatever it is the cell needs. And so our plasma membrane allows the cell to obtain from that interstitial fluid what it needs and exactly when it's needed. And then it keeps out everything else. It's a little bouncer. You know, it says exactly who can come in and who can't. And I'm like, and regulates it. Now, since it only allows certain things in and certain things out, we call it selectively permeable because it selects what it allows in. And the things that it wants in, some can pass through really easily through the membrane, and some do not, but we still need to get them in. So we have to figure out a way to get them in. There are two basic, groups of processes on how to get things to cross the membrane. Passive processes and active processes. A passive process means we do not need any ATP to move the molecule across the membrane. Now, generally why that's happening, substances are moving down their concentration gradient, meaning they are going from a high concentration to a low concentration. Things don't like to be crowded. Now, my example for this, and I had to change it because I've upgraded where I now have a freezer on the bottom. But when I used to have a freezer on the top, and I'm like, you know, and then I go shopping at Sam's Club, I would pack that freezer in, and I'm like, and pack it in, you know, rearrange it 5 million times so I can shut the door. Yay. However, what happens when I would still open it? Stuff would fall out. Now, it takes a lot of energy to shove all that stuff in and make it nice and tight and really high, you know, it's a highly concentrated freezer. It's got lots of stuff in it. And I'm like, but does it take any energy for the stuff to fall out? Nope, that happened. Things don't like to be concentrated. It's really easy to go from a high concentration to a low concentration. It's kind of the same thing what happens when you go to really big events and everyone's all crowded together. Everyone doesn't like it, and they try to spread it as far as they can, and you know, nice and easy. It's really hard to crunch things in together. It's easy to spread them out. And so when things are going from a high concentrated area to a low, they're like, oh, finally. And they just flow right on over there with no problem. No energy needed whatsoever. Now, active processes mean they need ATP. There are several reasons why it could need ATP. It only occurs in living cell membranes. We'll get into that later. But one of the main reasons is, yes, if you are going in reverse, like my freezer, if I take lots of stuff and I shove it into an area where it's already really concentrated, and I'm like, that can take a lot of energy to concentrate it in. Or maybe you guys have a closet like that at home. I don't know. But it takes a lot of energy to put things where there's already lots of things. That would be an example of when you might need an active process. It needs energy. It needs ATP. Now, there are different types of passive processes. And I'm like, again, none of these require ATP. They're moving from high concentration to low concentration. Kind of just depends on what molecule is moving. Now, and I think I've got pictures of these. And I'm like, I don't know if I want to talk. Hold on. Before I go into depth, then I'll be like, oh, look, I have a picture. Diffusion. And I'm like, there are 3 types of diffusion. Simple diffusion, carrier and channel mediated facilitated diffusion, which is a mouthful, and I'll explain it. That sounds way easier. Osmosis and filtration. None of these require any energy whatsoever. So we're going to look at the diffusion first. Now, diffusion in general is just molecules going from high concentration to low. Just before. They are moving down their concentration gradient, high to low. Now, I was going to say, I'm like, things that can change the speed of how they move. It can be the difference in concentration gradient. And I'm like, if you have a really high concentrated area and nothing, they are going to move really fast. If you've got like, I've got 10 molecules over here, and I've got 8 molecules over here. Is that a big difference? Shake your head. Between 10 and 8? No, and I'm like, what will happen? How many will move? 1 will move, because then the one side becomes 9 and the other one side becomes 9. I'm like, 1 will move, but that's not a huge difference. It's kind of like, oh yeah, I guess I could mosey on over there. If you have a big difference in concentration gradient, they're going to move a whole lot faster. Also, if the molecule is smaller, it can move a lot faster. And I'm like, so it can kind of, it can get squeezed through areas. It can wiggle its way a little faster if it's smaller. So molecule size can make it faster. And temperature. If the fluid is warmer, molecules move faster. And I'm like, kind of like with chemical reactions. The warmer it is, the better the chemical reaction. Same thing. The temperature can increase the speed. Now, I was going to say, I should have passive on here. Oh yeah, I was going to say, my first one, passive processes, but this is your simple diffusion. Molecules will passively diffuse through the membrane if it is lipid soluble and if it's small enough to fit through the membrane channels. Meaning, simple diffusion, it can go right across the plasma membrane. So when this thing says diffusion on the bottom of your picture, write simple diffusion. If it can go right through the membrane, there are some molecules that don't care that there's hydrophobic tails there. Some things do. And I'm like, and it can't go through there. But if it is small enough, it has no problem interacting with those tails. If it can go straight across the membrane with really not a lot of assistance whatsoever, it's called simple diffusion. Now, the facilitated, I was going to say the carrier or channel mediated facilitated diffusion, long. And I'm like, facilitated diffusion, the word facilitate means to help. That's what it means. If someone's a facilitator, you're like, I'm a facilitator. And you're like, cool. That just means you're helping somehow. So in facilitated diffusion, that means this molecule can't squeeze through there. It needs help. And it's going to use a protein. And so when we talk about carrier or channel mediated facilitated diffusion, a channel protein stays open nonstop. It's like a big old tunnel that never closes. And if you have a high concentration on one side and a low concentration on the other, they're just going to go right through. That's a channel mediated facilitated diffusion. These red things could not go through on their own. They have to go through a channel. There are some proteins that are specific for moving molecules across. This particular protein is like, all right, I like blue molecules and my job is to move them across. As long as I'm going from high concentration to low concentration, it's still a diffusion. We call this a carrier protein. It literally picks it up and carries it and then kind of spits it out on the other side. But these are facilitated diffusions. It just means they need a little help. Still from high to low concentration, Still no ATP required because we're still going from high to low. Now, this simple diffusion, examples of things that can go across, means, again, they don't care about the heads, they don't care about the tails, they can just get across. Oxygen can go across, carbon dioxide can go across, and fat-soluble vitamins. I'm not worried about those fatty acid tails. They can go right across the membrane without the use of any protein whatsoever. There's a picture out of your book. Some fat-soluble solutes, probably a vitamin, just goes right on through. You're facilitated, I already said it before, but there are certain molecules that can go across. They don't like the tails. They're all lipophobic. What's lipophobic mean? They don't like the lipids. It's like, who likes that anyways? Everyone's lipophobic. But if these are lipophobic, they don't like the tails. They just need some help. They need a facilitator, a helper. They need a protein, whether it's a channel or whether it's a carrier protein that picks it up specifically and moves it across. Here's the one out of your book. So, the carrier proteins, they are specific. Like this one is specific for little blue circles. This one is specific for green hexagons. And I'm like, the carrier ones are specific for what they move across. Now, osmosis. Everyone's like, I remember that in high school. Osmosis is moving of water across the membrane, still from a high concentration of water to a low concentration of water. Now, this does mix people up, because so far we've been talking about moving of solutes, like molecules and shapes of things. And I'm like, instead of moving of these molecules, I'm now physically moving the water. And it's still going from an area of high concentration to an area of low concentration of water. Water doesn't like to be crowded all by itself. It prefers to go where there is no water. Now, it needs to get across the membrane. But does water, you think it likes the fatty acid tails that are fats? No, these are hydrophobic. They hate water. Water hates it. The fats or oils and waters don't mix. And so it has to go through a channel. And so there would be a specific protein channel. And channel, because yes, it's fairly open. There's a little channel there. And I'm like, and this channel is called an aquaporin. Is this a protein for moving water? Because what's aqua mean? Water. And porin, it's like a pore, it's like a hole. And so it literally means like water hole. So they go through that. They're carrier proteins that move water. This only occurs when the water concentration is different. If you have a lot of water on one side of the cell compared to a lot of water on the other side of the cell. And I'm like, that water doesn't like to be crowded either. It will move too. So I'm like, here showing some of those water molecules, still going from a high concentration to low. They're just going to go through an aquaporin. They can't make it through that lipid bilayer. I'm going to preview this, but then we're going to review it next time too. Now, what can happen? I was going to say, I'm like, if I have this cool little tube that does this, if I put a membrane in it that allows molecules to move and water move, which side has more water? I should ask. Which side? What do you think? The left side has more water because there's less solutes in it, so this is more water. Which side has more solutes? The right side. Now, because this allows both of them to move across, if you let them sit there long enough, you'll have an even number of water on both sides and an even number of solutes on both sides. However, what can happen is if you put a membrane, water can squeeze through there, but those big red balls can't squeeze through that tiny little opening anymore. Where's the water going to want to go? It's going to want to go to the right because there was more water on this side, and so it wants to go where there's not as much water. But are the red molecules moving anymore? Nope. And so what can actually happen is you'll see water rise up in a tube. It's really cool. It's like you're defying gravity. This can cause problems because cells can swell and burst, or they can shrink. Neither is good. And I'm like, so keeping the same water concentration is extremely important. And I'm like, and I'll get into this next time, but would you ever give someone an IV of pure water? No. And I'm like, because if you have all these cells, are your cells pure water? No. Where's the water going to want to go? If you gave an IV of pure water, where will that water want to go? It'll want to go into the cell, because the cell doesn't have pure water. It's less than 100%, whatever it is. What's going to happen to the cell if all of a sudden water starts to go in and in and in? It's going to swell, and it can burst and die. That could kill someone. They don't like that. We'll talk about and look at pictures of all this happening next time, though. So we're going to end right there. Where's my thing?